AS Level Biology 9700, Paper 2, Variant 2, May-June 2025, question number 5. And question number 5 is, there is a global shortage of blood for transfusion. Researchers can culture bone marrow stem cells in the laboratory to manufacture a red blood cell. For potential use as an artificial blood product, the researchers collect bone marrow stem cells that are present in small quantities in blood rather than extracting them from bone marrow. So antibodies specific to bone marrow stem cells are attached to a tiny magnetic beads. The beads are added to a sample of blood and electric field is applied to immobilize the beads so that the bone marrow stem cells can be collected. So the antibodies are attached to the bead so that the sites used for binding to specific molecules on the bone marrow stem cells are left exposed. Name the term given to the specific molecules on the bone marrow stem cells that attach to the antibody binding sites. So the specific molecules on bone marrow stem cells that attach to the antibody binding sites are called antigens. They are antigens. So these are antigens are the molecules, often proteins or glycoproteins. They are protein or glycoprotein. So they are present on the cell surface membrane that can be recognized and bound by specific antibodies. So they are specific for one another. They have complementary with one another. So in this technique, the specific antigens on the stem cells are used as markers. So the antigens are also used as markers to selectively isolate them from the magnetic beads coated with complementary antibodies. So the name of those specific molecules are antigens. Now, next question is, suggest and explain the advantages of using bone marrow stem cells from the blood sample to manufacture artificial red blood cells. So, the advantages to use the artificial red blood cells. So, the answer could be the of 3 marks. The question is of 3 marks. There are so many different advantages of using. Actually, they are hemopoietic cells. Hemopoietic cells means that they can, uh, particularly, they can be uh, regenerate into different types of cells. So actually, hemopoietic cells are immature cells. So they can be used in the production of blood cell types. So these are specifically give rise to the blood cells. So they also act as multipotent as they can differentiate into many multiple cell types. So they can differentiate into red blood cells. They give gives a specific, you can say, uh, specific types of cells. So able to produce genetically identical cells produced through cell division being clones of the original. So carry out mitosis. So in this way, they are able to divide and carry out mitosis. So in this way, we can say that they have many specific advantages due to which they can be used as. So first of all, I would go with the first point, and according to this first point, they are hemopoietic in nature. Hemopoietic in nature. Due to hemopoietic in nature, they in nature, meaning they specifically give rise to blood cells. Second point can be they are multipotent in nature. They are multipotent, multipotent nature, so can can differentiate can differentiate into multiple cell types. Especially red blood cells. They have ability to proliferate, or they can increase their number, they can reproduce, they can replicate. So these are the many of the advantages to use them. And then I would like to go with another point, that they are less invasive, which means no surgical method is required. And immobilization method can only be used when they are in blood. So these are the easy method. Instead of extracting them from the bone marrow, instead of extracting them from bone marrow, it is easy to get them from the blood. So these are the few of the advantages according to the marking scheme and marks. These are enough. So we were moving to the next question, and next question is, one desirable feature of artificial blood products such as artificial red blood cells is that they should be economical to produce. Suggest other desirable features of artificial blood products should be. So, first uh, technique which I would like to discuss is that they have no need for, you can say, the product should be acellular. Should be acellular means free of red blood cell surface. Red blood cell surface antigens. This eliminates the need for cross matching, making it a universal donor. Then an artificial blood product should be shelf stable. It means it should not need to refrigerate. It should be, and the last point which I would like, it should be completely free of allergies or blood-borne pathogens. And then the product must effectively perform the primary function of red blood cells. That is the transportation of oxygen. So it was all about the question number 5. Please like, share, and subscribe my channel for more, to watch more videos on these papers. Thanks for watching.